hamster made your drugs? Yes, there are loads of drugs on the market that were made from Chinese hamster ovary cells. In 1956, these cells were found to grow so quickly that scientists realized they'd be great for making proteins, aka drugs that will make you a butt ton of money. So, these cells were genetically modified to produce a target protein, or more specifically, a monoclonal antibody that can be used to treat cancer, infectious disease, and autoimmune issues. Now, let's look at the first steps required in manufacturing of these drugs, which industry defines as upstream. First, you take your frozen genetically modified cells, thaw them in a water bath, then pipette them into a flask containing media, which supplies nutrients for your cells to consume and grow. Starting with a small flask, you place it in an incubator to control temperature and CO2 levels. Let the cells multiply for a few days, then transfer to a larger flask with new media and incubate for another 3 to 4 days. This process is repeated until you have a large enough number of cells to transfer to a wave rocker. A wave rocker is exactly how it sounds. A bag containing the culture is placed on a tray that continuously rocks side to side. This rocking motion allows for gas transfer between the solution and the surrounding air, allowing the oxygen concentration of the solution to be controlled steadily at around 30 to 40% and CO2 at 5 to 7%. After a few days of growth in the wave rocker, it is time to transfer to the first seed bioreactor. A bioreactor is simply a fancy fermenter, kind of how you make beer with a few more controls. You want to control temperature, pH, dissolved oxygen, CO2, and constantly be measuring your viable cell density. The smallest bioreactor, typically around 60 liters, will be inoculated from a wave bag. The cells will grow for 3 to 4 days, then a full or partial transfer is made to a larger bioreactor supplied with fresh media. This growth and transfer process will be repeated until the target volume is reached, which can be as much as 20,000 liters. Next, that cell media solution is sent to a centrifuge. Because CHO cells make the target protein and secrete it outside of the cell, and because the protein has a low density compared to the cells will move to the outside and bottom of the centrifuge, while the protein will move towards the middle and up the centrifuge, which you then call your centrate. At this point, you have separated a lot of the gunk from your target protein, but there's still a lot of impurities in the solution that would harm you if you injected it. So that's when filtering comes in. In industry, this is called downstream processing, which will be covered in a later video.